Hello, everyone. Greetings from JDRF headquarters here in New York, and welcome to the October RIV call, which is focused on cell therapies. My name is Jaime Giraldo. I'm Associate Director of Research here at JDRF, and in that role, I lead the Cell Therapy Project, which focuses on development of therapies to transplant insulin-producing cells. My background is in bioengineering with a focus on tissue engineering, and prior to joining JDRF, I was down at the Diabetes Research Institute at the University of Miami, focusing on differentiation of stem cells into insulin-producing cells and methods to protect the cells following implantation. So as I mentioned, today's focus is going to be on cell therapies, which is part of the CURES program. Now, when we speak about cell therapies here at JDRF, we are talking about therapies which deliver live cells to treat or reverse disease, in this case, particularly insulin-producing cells, to replace the loss of functional pancreatic islets or beta cells in the islets. And so the goal here is to restore insulin independence and blood glucose control, and so we are focusing on accelerating development of cell therapies by supporting research and preclinical and clinical testing, by supporting activities for regulatory approval, and of course, improving access to these therapies. Now, the current state in cell therapy, we have two main focus areas, which is the cell source and protecting those cells from the immune response. In terms of the cell source, we now have multiple sources of cells, which had been or are being developed by different companies, which are making their way into clinical testing in terms of people with T1D. So our main focus right now has shifted towards cell protection. How do we protect these cells from the immune system? Now, one of the caveats of cell therapy is that they require a lifetime of broad immunosuppressive therapies, which carries risks for the people taking those therapies, and so limits the population of people that are eligible to receive a transplant. So it's not scalable for the broader T1D population because it's limited to people that have severe hypoglycemia unawareness, severe hypoglycemic events, and are having a very hard time managing their diabetes. And so we need to find ways to deliver these cells and protect them from this immune response without the use of these immunosuppressive drugs, and so this is our current focus right now with the JDRF cell therapy program. Now, we're looking at multiple approaches to avoid this immune-mediated destruction of beta cells. The one that most people are familiar with, which we have been supporting for a while, is encapsulation, where you have a device that basically creates a physical barrier between the cells, the insulin-producing cells, and the immune system of the host, and so that way the cells can't attack the implanted cells. The immune cells can't attack the implanted cells. Now, there are some challenges with those devices. These are foreign materials that are implanted into the body, and so the body does recognize them and react to them, which creates this kind of fibrotic response. There are challenges in terms of nutrient delivery because the cells don't have access to the circulatory system and blood vessels directly, and so it's hard for them to get nutrients, oxygen, and sense the glucose in the blood and secrete insulin accordingly. And so we do have challenges delivering a large dose of cells that's going to have a therapeutic effect. And so we're looking at both macroencapsulation devices, which are large devices to deliver a large dose of cells, which makes them easy to retrieve, but have these nutrient delivery issues, and also at microcapsules, which are smaller capsules, which enhance the delivery of nutrients, but then are a bit more challenging to retrieve. We're also looking at scaffolds, and so scaffolds are a really cool technology because it kind of helps sparse out the cells in space, and so that way they're not all clumped together competing for nutrients, and so it helps with their survival and function. And it also presents us an opportunity to engineer the site where these cells are being implanted, so they can be used to deliver different drugs, deliver oxygen, or deliver factors that promote the ingrowth of blood vessels. And so that way we can kind of modulate the environment where the cells are to ensure that they survive and function. Now, of course, these are open scaffolds, and so they don't provide immune protection. So we need to incorporate strategies for immune protection. So that can be achieved through local delivery of drugs or through some of these other approaches you see on the bottom here, which include immune modulation, which is local delivery of drugs to alter the immune response, or perhaps even looking at reprogramming that immune response so that the immune system doesn't sense these cells as foreign and attack them, or also gene editing methods that could be used where we basically use new technologies such as CRISPR to edit the genes of the beta cells that are being implanted so that they don't express markers used by the immune system for recognition. And so basically these cells become cloaked or immune evasive, and so they evade the immune system, and so they don't cause that type of immune reaction and don't get attacked. And so we have multiple labs, multiple academics at different universities and companies working on one or combinations of these approaches and these strategies. And so we're making sure we support them to ensure effective translations of these technologies into products that can be commercialized and reach the clinic. In terms of specific JDRF projects we wanted to update you on today, we do have a very exciting project ongoing, and this is a partnership with ARMI, the Advanced Regenerative Manufacturing Institute up in New Hampshire. And this is focused on translating a manual protocol that Dr. Jeff Millman, who you see here at Washington University, has developed to differentiate stem cells into insulin-producing cells, translate that into kind of an automated, scaled-up process so that we can make these cells in a very reproducible manner and at large scales so that we can distribute them to other investigators in the field to accelerate the development of these technologies that I was just speaking about earlier. So this will be very useful for the community so they can test their different technologies with a standardized kind of cell source, and it'll allow us to compare different technologies side by side, basically comparing apples to apples. And so this grant is $2 million. We recently also funded another grant between Dr. Millman and ARMI, focusing on protocols for iPSC-derived stem cell lines. And so one of the challenges with differentiating stem cells into insulin-producing cells is that these protocols have been optimized with specific cell lines, which are used in the lab, and they don't always translate to other cell lines. So you can't use the same protocol to differentiate multiple cell lines into beta cells. They don't all respond the same. And so this grant is going to be focusing on how we can make adjustments to these protocols so they are more robust and work on multiple cell lines of induced pluripotent stem cells, or iPS cells. And this could be really interesting because in the future we can derive these cells from individual patients, basically making this an autologous transplant, so cells derived from the same patient turned into beta cells and re-implanted into that patient. And so that's another very exciting grant that we funded recently, and we're looking forward to seeing the results from this effort. Another very exciting project that we funded recently is with a partnership with Aspect Biosystems, which is a company that focuses on 3D bioprinting. And so Aspect, what is unique about their technology is that they have this novel microfluidic print head, which they use on their 3D bioprinting system. And they're using this to integrate different biomaterials and cells to create these implants that are laden with insulin-producing cells. And so the idea is these implants are designed and optimized to maximize cell survival and function. And of course, the materials are selected to do that and to achieve immune protection. And so they're working on this really cool technology. We're looking forward to also seeing how this partnership progresses. And so the aims are to test, optimize, and develop this implantable product and move it into IND-enabling studies and hopefully into the clinic in the near future. Now, of course, the cell therapy project is involved in quite a bit of these centers of excellence, the JDRF centers of excellence that have been established, which is really cool. We have these different collaborative teams across the country working on very important and challenging issues that can have a big impact here in the development of these therapies. As you can see here, we have 4 the University of Michigan, which is focusing on making a better source of beta cells by increasing their energetic and metabolic capacity. So they're more robust cells and don't suffer as much in adverse environments. The Northern California Center of Excellence, which is working on immune mechanisms of T1D, the interface between immune cells and beta cells, and gene editing of T cells or immune cells, which could result in enhanced survival or immune protection of the transplanted beta cells. So we're teaching the immune system how to tolerate beta cells here. The New England Center of Excellence is also working on gene editing, but their focus is more on gene editing the beta cells themselves. And so how do we make these cells less immunogenic so that the immune system doesn't react to them as strongly? And finally, the Center of Excellence at the University of British Columbia, which is focusing on developments for new sources of insulin-producing cells and making the manufacturing of these cells cost-effective. They're also working on protecting the beta cells from immune attack and focusing on and monitoring beta cell stress. So of course, we want to avoid beta cell stress. That'll enhance the survival and function of the cells following implantation. Now, some recent funding opportunities that we've released and new areas where we funded projects include immune engineering. We had an immune engineering RFA. As I mentioned before, we need to protect these cells from immune rejection without the use of broad immunosuppression. And so while encapsulation is one important solution, challenges remain with that approach. And so we need to pursue this challenge from multiple angles, immune engineering being one of them. And this kind of tags into the immune modulation area that I spoke about earlier. So the idea here is to catalyze the development of immune engineering strategies to modulate immune responses to implanted cells. And what immune engineering means is basically combining biomaterials in different drug delivery systems that have been developed in the bioengineering area with novel drugs or novel checkpoint inhibitors or different targets that the immunology field has been working on more recently. And so trying to combine these two basically areas of research to come up with these really novel and impactful technologies that could lead to protection of the implanted cells without the use of broad immunosuppression. And so we funded about a total of $2.9 million in this area, and we're really looking forward to seeing the results and the findings from those studies. Now, this year we launched a immune imaging RFA, and so this is also very exciting. Currently the methods that are used to analyze the efficacy of these different methods for immune protection of the cells are inefficient in terms of the time and cost, and they do provide very limited information. So typically you do different experiments where you have several animals, you have to check them at different time points, where you have to process samples, and it does take quite a bit of time and energy. And so the idea here is to leverage advances that have been going on in medical imaging, which are applied for cancer and inflammatory disease, and apply those to T1D and the development of these new immune modulating therapies, so that we can accelerate the analysis of how efficacious they are, and really try to move things faster along the development pipeline, identify what's working, what's not working, rather quickly, and then move on so we can accelerate the development of these therapies. So we put out this RFA. We've received LOIs. Full proposals are due quite soon. And so we're really excited about the potential for this area to really accelerate the development of alternate methods for immune protection of the cells. And so we're expecting to fund about $4 million of research in this area, but of course that's still to be determined, given that we still have to send out the full proposals for review and see what the outcome of the review is. But we're very hopeful that we'll get some really interesting projects from this RFA. And so cell therapies have been making quite a bit of progress. I think we've all heard about the first results of the stem cell derived islet transplant trial, the VX-880 trial from Vertex. If you recall, the subject in this study, which is Mr. Shelton, or patient 1 here, prior to the procedure, had been taking insulin for 40 years. He was constantly monitoring blood glucose. He had 5 severe incidents in the year prior to receiving the transplant and had no detectable insulin production. Following this intraportal infusion of stem cell-derived islets, now he does not require exogenous insulin. His body basically regulates his blood glucose automatically, but he does have to take chronic immunosuppression therapy. So it's very promising, but again, this is why it's so important for us to develop these other alternative methods for immune protection to obviate the need here for chronic immunosuppression and really make this therapy available for anyone that has type 1 diabetes. But it's very exciting. It's incredible to see that we have a stem cell-derived product in the clinic that is working. Now, ViaCyte has also made significant advances. As you all know, you're familiar with the PEC and CAP, or the VCO1 trial, which demonstrated that stem cells from their facilities can differentiate into insulin-producing cells and produce insulin after implanting them into people. They've demonstrated long-term survival of the implants and no adverse effects. And of course, their PEC Direct product, which is also in clinical testing, which enables vascularization of the device and does require immunosuppression, also has yielded some promising results. Now, we're also really excited about the initiation of a clinical trial with gene-edited stem cells, which is in partnership with CRISPR Therapeutics. And so, again, as I mentioned earlier, the idea here is to edit the beta cells that are being implanted so they do evade the immune system. They don't express these surface markers that the immune system recognizes, and so that way the cells can evade the immune system. So JDRF, of course, had a partnership with ViaCyte for over a decade. We played a critical role in the establishment of these different cell lines and protocols for differentiation of the cells, which we're very proud of. And of course, we also advocate for CIRM, which is the California Institute of Regenerative Medicine, which also supported ViaCyte for quite some time. Now, I think all people also have heard the news about Vertex acquiring ViaCyte, and we've been getting quite a few questions about this. What is the impact on the research, on the field, on our strategy, pros and cons? And so just to highlight and address some of those points here, ViaCyte has 15 years of experience in this space, so they've learned quite a bit in that time. They have a track record of working with regulators to move a lot of their studies into the clinic, which is invaluable expertise. They have years of human data that's incredibly valuable in terms of trying to figure out how do we implant the cells in different sites, etc. And they do have multiple cell lines that they're working on, and they've worked out the logistics in terms of manufacturing these therapies, shipping them to different clinical sites, etc. And so ViaCyte has a lot of strengths. Of course, Vertex has a lot of strengths as well. They have a strategy to pursue future generations of products with iterative functional improvements, moving from using immunosuppression, encapsulation, gene editing, etc. They have the first proof of concept with the stem cell-derived islet that demonstrated safety and efficacy. And of course, they have a successful development and business model in non-T1D indications. So Vertex is a huge pharmaceutical company. They have a lot of experience in bringing drugs forward through clinical trials and FDA approval. And so they have a lot of expertise and infrastructure to make these therapies a reality. And so I think the combination of these two, of course, is I think a strength, because they're going to be able to leverage the expertise and strengths of each of them to really accelerate development here. Some questions, and the answers remain to be determined, are what are the next steps for removing immunosuppression? As I spoke to earlier, there are a lot of options that are being pursued, and so what that actually looks like moving forward is still to be determined. We also don't know exactly how they will work with JDRF and other companies in the field, and then also how they will work with and compare to other large companies in this field. So that remains to be determined, but we are very hopeful and optimistic about the future. We think this is a very, very good development, and so we're looking forward to the rest of the results of those trials and other studies that are coming up soon. So, in summary, basically, we think that Vertex acquires ViaCyte, 1 + 1 is greater than 2 here. This makes the field stronger and creates a sense of urgency, and is going to accelerate the development of cell therapies. And really, it does not directly affect the cell therapy project area's strategy, roadmap, priorities, or portfolio. We had already accounted for both of these players in the space and their activities and what they were doing within our strategy. And so now that they are basically working together, they're still pursuing the same things they were pursuing before. So, we are still focused on the different priorities we've identified to make sure that we accelerate development of these therapies. Now, what is the role of JDRF in catalyzing all this activity and moving the ball? So JDRF does fund a large portfolio of grants in this space. Currently, the active portfolio is about $70 million, as you can see here. And so the breakdown of the portfolio, you have a large percentage of projects that are in discovery, early discovery, a majority of the portfolio here, which is in early preclinical testing. We are hoping to move those projects along the development pipeline into late preclinical testing. You can see here in the light gray where we have some projects already there, and then ultimately move those into clinical testing, which is the light blue you see here. So we do have projects across the entire development pipeline here, and we're constantly working to move those projects along the pipeline towards the clinic and towards commercial therapies. In terms of the distribution of the portfolio across different areas, you can see that in this diagram. Cell source is only 3%, and that goes back to what I said earlier about we already have multiple companies that are able to make stem cell-derived insulin-producing cells. There's a lot of investment in industry in this area, so we think we can shift our focus away from that and more towards how do we deliver the cells and protect them from the immune system. And so encapsulation, which is an area that we've invested in historically, is at about 25% of the portfolio at this time. And graft survival, vascularization, and oxygen is about 24%. And so this is important, making sure that the cells survive once we implant them, regardless of the immune response, just in terms of getting enough nutrients and being able to survive the procedure. And here, the other section, the light gray, which is alternative methods to immune protection, is 40%. So as I mentioned, we've invested in encapsulation for some time. There are challenges, and so we want to make sure we're exploring all the options out there, and so attacking this problem from multiple angles. We've also focused a lot on the investments in alternative methods for immune protection, which includes immune modulation and gene editing. Now, the last part, which is the light blue here, it's accelerating clinical translation, and this is just supporting different cores or different technologies that we can use to accelerate translation into the clinic, basically. So, for example, the imaging RFA would fall under this category because it's going to accelerate the assessment of different therapies in development and hopefully accelerate the translation of successful ones into the clinic. I'll speak to just the beta cell replacement consortium, which is the consortium JDRF has set up from all our funded investigators, which include people from over 20 or 30 institutions. It has about 50 or 60 members, and it's really bringing people from multiple different disciplines because it is such a tough challenge, and we need expertise from different areas, including animal models, transplantation, immunology, biomaterials, bioengineering. And so we want to make sure we bring all these people together to talk about the challenges in their areas and how it applies to beta cell replacement in type 1 diabetes and the opportunities, and hopefully foster some collaborations that will accelerate the development of these therapies. And so members of the consortium identify and test novel biomaterials, they design better devices, capsules, and scaffolds, they standardize the data and the methods to really have consistent results and be able to transfer technology between different labs, translate the results from small animal models towards large animals and ultimately humans, and optimize different approaches for local immune suppression. And so JDRF really has been a leader in this space. This is just a kind of a highlight or overview of the program and its history, which was first established in 2008, and back then it used to be called the Encapsulation or Beta Cell Replacement Program. The first consortium meeting was held in 2013. In 2014, we already saw clinical trials starting for 2 encapsulation beta cell replacement therapies, and we saw the successful differentiation of stem cells into beta cells. In 2015 and 16, the encapsulation consortium expands, so it's no longer just encapsulation, but focusing on all of these other methods for immune protection, focused on novel implantation sites and new biomaterials. And now to the more recent years, 2018 to 2021, we've updated the consortium name to the Beta Cell Replacement Consortium, and we have all the exciting clinical data from the Vertex, ViaCyte, and Cernova clinical trials, which we're very excited about. Now, how does JDRF accelerate cell therapies? I think this is a prime example of how JDRF does that. We first funded Doug Melton at Harvard to create stem cell-derived insulin-producing cells in 2000, and he's been working on that ever since, finally succeeding in 2014. In 2015, he established Sema Therapeutics, which wanted to turn these cells into a therapy and a product. In 2017, the T1D Fund, which is JDRF's venture philanthropy arm, makes a catalytic investment in Sema, in this company, which is subsequently acquired by Vertex. In 2021, the VX-880, which is Vertex's cell therapy, receives Fast Track designation by the FDA. And now in 2022, we see the data from the first person to receive this therapy, and they no longer require insulin. So from 2000 to 2022, JDRF played a huge role in all of these developments by really seeding the support for this research, seeding this research, supporting the development of the company, which ultimately resulted in the acquisition by Vertex and the results in the clinical study. There are clinical trials ongoing aside from the Vertex and ViaCyte studies, which are listed here. As I mentioned, Cernova, which is a company based in Canada, is working on a scaffold type of approach, their cell pouch, to deliver the cells in the subcutaneous space under the skin. So the Vertex trial does involve portal infusion of cells, so infusion into the liver, and that has its own challenges, and it's not an ideal site. So we're exploring alternative sites such as the subcutaneous space for delivery of the cells. The team at UCSF in San Francisco are looking at co-transplantation of parathyroid gland with islets, and this is because parathyroid glands appear to induce a robust vascularization of the graft and the site, and so that will enhance survival of the cells and function. There are quite a few trials going on at the University of Alberta in Canada, where they're exploring polyclonal Tregs to protect the implanted beta cells, anti-CD40 ligand immunotherapy, which is an alternative to immunosuppression, which might not be as toxic and more effective, and also a deviceless site, basically a similar approach in terms of the implantation in the subcutaneous space or the skin. Finally, more recently, one really exciting project that we're looking forward to seeing the results is a trial at the University of Adelaide in Australia, and this is co-funded between JDRF here in New York and JDRF Australia, our affiliate there, on BTM for implantation of islets in the skin. Now, BTM is a scaffold device that's been used, it's approved, and it's used for treating burn wounds, to basically regenerate the skin and promote vascularization. And so we're trying to apply that here for the implantation of islets in the skin, and so we're hopeful that this will show some promising results. So you can log on our Clinical Trial Connection website to look for other clinical trials in this space in general. So in summary, clinical islet transplantation has demonstrated that beta cell replacement therapy can provide a functional cure for type 1 diabetes, but it's limited by the scarcity of donor tissue or donor islets and the reliance on broad immunosuppression, which is why we need these alternative renewable sources of beta cells such as stem cells, and figure out methods to protect the cells without immunosuppression. So as I mentioned, we now have renewable sources of cells, stem cell-derived beta cells, which are being tested in clinical trials, and several companies are making these cells. And so our current focus has shifted to ensure that we have methods that can deliver the cells so that they remain viable and functional, and methods to protect them from immune rejection without the use of broad immunosuppression. So we are supporting research exploring alternative sites of transplantation, such as the skin or the omentum, and methods to deliver oxygen and enhance the vascularization of the cells. And we're exploring multiple avenues to protect the cells from the immune response, such as encapsulation, immune modulation, and genetic modification of various cells. We continue catalyzing progress, so we continue having the consortium meetings and making sure that people are exchanging ideas and collaborating and exchanging data and results, both from academic institutions as well as industry. JDRF investment has led to significant additional investment from the private sector to develop these technologies, and we continue to work with companies and regulators to facilitate a path for regulatory approval of these therapies. So multiple clinical trials are ongoing, and so we are very excited and looking forward to the next few years, because we are expecting some very interesting findings, and so moving the field forward will get us close to a cure, hopefully sooner rather than later. So with that, that's the end of the presentation. I look forward to your questions next week, and hope you all have a great day. Thanks. Bye.